Yeah. Good evening to one and all. I welcome you all to this faculty development program organized jointly by Center for Ocean Research and Department of Biotech, association with the Earth Science and Technology Cell, established for marine biotechnological studies. Today we'll be it's organized by Earth Science and Technology Cell in association with marine biotechnology studies, funded by Ministry of Earth Sciences, Government of India. And today we'll be having this lecture on the title Nanomaterials, Synthesis and Application from Marine Sources. In this particular topic, we are just involved in synthesis of various nanomaterials using Sargassum species, red algae, and we have also isolated very marine bacteria from seaweed species, and we have used the same for reduction of many no metals. And based on that, we have just characterized them and we just published the research works also. We just used some for some biomedical applications also. In this lecture, I have the part of our research work and other research which are involving outside, apart from our center, outside the other imaginary research contributions also, which will be just giving an idea about how to use sources to synthesize the nanomaterials and some important applications which are in progress. And before that, I'll just give a small introduction about Center for Ocean Research. Center for Ocean Research was established in the year 2007. Here, our major thrust areas are mariculture and fisheries, marine biotechnology, ecology, and microbial technology. One of research group of mycotoxicology, biocorrosion, and we are also associated with entrepreneurship related programs, and they're also involved in patent products and education and training. These are the facilities which are available at Center for Ocean Research exclusively. We have ICP-MS, GCMS, 2D electrophoresis, stereo zoom microscope, FTIR, UV-Vis spectroscopy, all fundamental and all advanced instruments which are related to marine technological studies. And we do have biosafety cabinet for biomedical applications, and we also have CO2 incubator saline studies later. We do have photobioreactor and biofermenter, and we also have this muffle furnace, and this is some facilities which are available at the Center for Ocean Research. And regarding the funded projects, we have nearly 43 funded projects. We have nearly 11 projects from DST. It is from PDP Nanomission, and NSTDB, ECR, TAR, and the DBT also. We have nearly four projects: ICR, AARB, Ministry of Earth Sciences, DRDO, IFS, in BRNS, in ICMR, DST, CRB, and UGC NPDF also. We all have these funded projects in our center. It was a past journey of nearly 15 years. And we do have many funded programs also. And as far as publications are concerned, we have which are established from scientists research groups, and they are also indexed in SCI journals. And we have nearly a citation of 4,600, and our H index is 33 for center alone. And in university H index, we contribute nearly 13 out of the 73 in Scopus. And we had a major inauguration of the mega facilities internal research park of Center for Ocean Research in February 2005, in the august presence of Secretary Minister of Earth Science, Founder Chancellor Sir, Sir, and all were involved during we invited during inauguration. And this facility was established in the 2015 research park. We also have a marine biotechnological exclusive study, and it was totally funded for nearly 4 crore rupees for marine biotechnology study. It was inaugurated last 2 years ago, and the project is presently in progress. And our ocean research team is also involved in the MSME TIFAC academic partnership program. And these are some other research activities which are in progress. And we have our center is recognized by DST FIST under the Earth Science category based on our research publications under funded projects which we have carried out successfully. And we also closely work with entrepreneurship activities. We have established technology business incubator, and in India, it is one among the specifically established for marine bioresource utilization. And we also running the programs which are related to DBT, skill development programs, special products. These are we have completed this 3 years program successfully, and these are the funding agencies which we had the projects. And now we'll move on to the topic. The word nanotechnology, as we all know, it's just bringing the size down, bringing the particles, bringing the materials and trying to find out how the structural properties or how the optical and electronic properties are varying from the bulk metal to the normal metal. And we can generally call it is a science and engineering to create materials between 1 to 100 nanometer. And in this case, if we just see anything which follows below this or which is having the size more than 100 nanometers, the particle size varies. So when you just synthesize a gold or silver, we have to just see to that it is being less than 100 nanometer for the specified applications which we are just arriving, or for the specified application, if it be a coating study or it can be a biomedical study, whatever it is, the study, if the particle size is less than 100 nanometers, the application oriented regarding which we are just focusing will be a better thing. And nanostructure thin films are used in solar and coating applications. This which we just take it as for in case of nanotechnology. A decade ago, the efficiency of solar energy harvesting was nearly 12 to 15% only. Presently, we harvest nearly 47% of solar energy by using the advanced thin film coating techniques and increasing the efficiency. We're just talking about the efficiency. 12% efficiency in harvesting the solar energy has increased to 47% due to the size reduction and optical properties. As far as marine biopolymer is concerned, the chitin and chitosan, those biopolymers, which is trying to make up with utilized in biomedical applications. And in inorganic nanomaterials, they are leading high dielectric constant, as we know, the band gap and the energy applications wherever we are just using them. And this is the nanomaterials. Faraday researched in the 1857 and in 1958, Richard Feynman explained that there is plenty of room at the bottom, where which mean to say you just go down the particle size from top to bottom. If you reverse this both, he just said if you just come down the properties, if you just come down the size of the particles, you can visualize that the optical and electronic property of a metal is not the same, which is what's behaving at the micro level, and so it is entirely different. So from Richard Feynman, the term was moving forward in 1974 as nanotechnology, which he meant to say that any material which is at 10 power minus 9 meter can act as a nanomaterial, and these materials are having the properties which can be engineered, or you can understand in this fashion. You can coat it on a surface. You can make it for an application where which a bulk material can do the application. Same properties can be existed on coating a thin film or modifying it with a stabilizing agent, or if you are just coating it on the agent, all this can be carried. So this term of engineering this nanoparticle came into conventional from 1974. And this is the simple understanding about what is the top-down approach and what is the bottom-up approach. If you just take a metal to its smallest size, if you just try to bring down, there are various techniques. For example, if you just take a ball milling, or if you just take a mechanical milling, or you try to down the nanomaterial in the bulk form to its lowest form. So we just call it as a top-down approach. In this, if you just it is not oriented to charge, or it is not oriented to any alloy or anything, the bulk material can be just grinded down. But in case of bottom-up approach, you can just build up a molecule. You can just build up an atom from its lower level to next higher level. In case, for example, let me explain. You just visualize a gold atom. It is very small. If you just go to a palladium or a platinum, it is in the size of a gold atom is in the range of picometer only. So you can just visualize picometer is 10 power minus 12 meter, whereas a nanomaterial is 10 power minus 9 meter. So beyond this, the nano atoms are small. We are just trying to arrange those atoms for our selective applications, where we can just find out that a 10 power minus 12 meter particle or an atom is being to make a nanomaterial which is in 10 power minus 9 meters. So you can just visualize, so a gold atom is smaller than a nanomaterial. So reduction of size is related one to property and the surface area which can be applicable to bring the materials to the next level. So the atom is very small. The nanomaterial is bigger than that. Your atom of gold or palladium or platinum are all lying in the picometer range only. So we're just trying to bring it to the nano level for our desired application. It can be a nuclei and it can be growth. It can be molecular level. It can
Ganesh,
be. Yes.
Sorry, sir.
Yes.
Actually, your voice is clear. Even we are recording the session, so if it is not comfortable, even you can close your cam and you can talk.
Is it audible, sir?
You just proceed, sir.
Is it better?
Sir, yeah, please continue, sir. You can continue.
Yeah. First, this is the size of the atom for bottom up approach. You can just from the palladium atom is 163 picometer. So these are the various sizes. So in this, you can just arrange 2000 gold atoms in a cell. This is what the understanding of the atoms or designing a nanoparticle means. So that's why we are just talking about 1 to 100 nanometers. If you just go below 2 nanometer or if you go below 3 nanometer, the particle will become amorphous. So you can't
sorry for the inconvenience. I think he might have lost. Sir, we have lost your PPT, sir. Can I, sir? Can I, sir? Can you hear me, sir? Yeah. So please unmute yourself, sir. Yeah, yes.
This is the size. This is the arrangement. This is the arrangement of the atoms which are just trying to bring to the nano level.
There, sir.
Yeah.
Yes.
Moving now.
Yes, sir. Yes,
Yeah.
sir.
Yeah, fine. Sorry. In this metal nanoparticles, I just it is a image taken from collection surface B article. This is a simple presentation to just show how the bioreduction occurs. And if we just go for the materials which we have carried out or any common reduction, it is just a change of color or it is just a change of surface of a material. Okay. For example, let me just explain with the gold. If we just take gold, we just have it as in the form of auric chloride. If we just record an UV, you can see that the nanomaterial, the auric chloride at this oxidation state of 3+ will be just having a slight yellow color. And in UV, you just know it is not dependent on concentration. So if you just come down, you can just see you will be getting a peak at 390 or 380 nanometers. If you just record a UV. Once you use a reductant, you just talk about chemical reductant. If you're just using trisodium citrate, you're just going to reduce it. You can see that nanomaterial formed or a nanoparticle formed will be having the wavelength at 520 nanometer. to just have a small memory, gold will have its surface plasmon resonance at 520 nanometer, and silver will have its surface plasmon resonance as 420 nanometer. So if it is going to be plus or minus, let us not bother, since we just know that there can be various types of error. It can be an instrumental error, it can be an intermediate error, it can be a chemical error. So plus or minus 20 nanometer differentiation or change in wavelength is permitted to just understand that the reduction has completely taken place. We are in this particular study. So the biosynthesis is having a small comparison better than the chemical synthesis. There is an advantage also, there is a disadvantage also. The main advantages are it's a colloidal stable particles are forming, it is reproducible, and adaptable surface properties are there, and it's easy and cheap. That is what the basic understanding is. We're just trying to make a culture, we're just trying to make a reactor, or if we just don't use a seed species or anything, it's easy and cheap and non-toxic chemical, which means that a level of which is existing will be less compared to a chemical synthesis, and environmentally benign solvents are being used, and renewable materials also there. Okay, I'll just give a small piece about the characterization techniques of nanomaterials. And here, UV-Vis spectroscopy. Fundamentally, if we just synthesize nanomaterial, if we just use a precursor, silver nitrate or palladium chloride or chloroauric acid, whatever it is, if we just use a precursor and if we just use a reductant, let us not directly, not necessary, it is not essential to go to the level of them and go record, yes, I have just got an apple. No, if you just record, you can just find out that the particles with the precursor will be showing you and reductant will be showing you a different. And in spite of this, basic visualization will be the color. The nanoparticles will be always ruby red in color. The slight variation, as I said, it can be based on the, or it can be based on the surface changes occurring in the molecule. So the plasma resonance, we can just record it using spectroscopy. And since we're just talking about the bio
Ganesh sir, we have lost the presentation again, sir, actually. Can you share again? Yeah, yes, sir. Yeah, we can see the slides now, sir. So please unmute yourself. So please unmute yourself, sir. Yes. Yes. Yes, sir. Yes.
Or specialties where you can just identify the particle and surface morphology, even atomic force microscopy to study surface topology. And DLS and zeta potential, we can just find out what is the type of charge which is existing in the nanoparticle which are synthesized. For example, with you can find out in case of XPS, you can just determine the chemical. For example, if I just to be more clear, if just taking a hexavalent chromium, if you are just having an energy state, if it has been reached, if it has just moved to its oxidation state, and the oxidation state has varied from 6+ to 3+, you can identify using XPS. Okay, your inductive plasma here, you can just using ICMS, even if you have you can determine the concentration of particles which are present in the given solution or the solution which you have synthesized. Okay, GCMS is just to use or to identify the chemical molecule which is attached on the surface of your nanomaterial. It can be a gold or silver. Based on the reductant, we can just find out. And this matrix MALDI mass, using this MALDI mass, you can just identify the total molecular weight of the polymer, or if bulk molecules are involved, you can just identify the molecular weight, higher molecular weight using this Maldi-Mass technique. Okay. Now we just move to the synthesis of materials in the marine resources. Marine resources, we just synthesize using algae, bacteria, fungi, biopolymers, sponges and crustacean, mangroves and other sources. These are the various marine sources using which they are synthesized the nanomaterials in different parts of the research institutions, in various other research institutions also. We have just synthesized nanomaterials using algae, fungi and biopolymers, and even sponges also. I will just explain a few of the research work which I have just shown. Why marine resources? This chemical molecule or chemical compound which is present in a seaweed withstands extreme variations in change in pressure, salinity, pH, and temperature. So the chemicals and metabolites produced are having unique in its biodiversity and its structural and functional features. So marine resources are expected to have better properties based on its stability. Okay, and needs more explanation. This reason is we know that alkaloids are present in terrestrial plants and seaweeds, but first alkaloid was identified in the year 1805. But first alkaloid in a marine algae was reported only in 1969. So we can just correlate these two differences. So the necessity for research, so the necessity for exploring molecules has a big variation. So there is a wide possibilities of various new research areas, and identifying of new research molecules can be done using this marine environment. And as far as this fungi is concerned, marine fungi also Penicillium, Aspergillus, Fusarium, all this can be explored much, where we can identify new chemical properties, new chemical metabolites, which will be helpful. The reaction parameters in the synthesis of nanomaterials. In order to decide the size, shape, and stability of the nanoparticles, generally in synthesis, the following maintain a pH. We have to optimize the temperature, either if it's a room temperature or you are just going to reflex a mixture. Time intervals based on the kinetics also, your experience can vary. Reactant concentration in the synthesis. If you're just taking an extract, it can be optimized in various microliters to achieve the desired particle, and exposure to light resources. I'll just explain a small point here, one more point. You are just, let us imagine that we have synthesized a nanomaterial of 60 nanometer. It is 100 ml, it is kept in a 100 ml beaker. And if you are having a 60 nanometer particle size of 100 ml in just a conical flask, you just add 10 microliters of sodium chloride in it, a dissolved solution. You can see to that the 60 nanometer immediately moves to the higher level. It can be 80 or 90, but you can't find that the 60 nanometer again if you record a TEM or if you just record a data potential of particle size, whatever it is. This shows the nucleation varies even based on the small factors. So the reactant concentration, the synthesis, and its exposure plays a major role. This is how the synthesis of various algae are involved. And I just said this characterization also. This is taken from the source manuals. There are various applications. Antibacterial applications are there, antifungal, anti-cancer studies, anti-fouling studies, bioremediation studies. There are various applications from the marine-derived nanomaterials are being presently, the experiment results are just giving positive. Okay. Sargassum, this marine algae, it's a brown algae. We just used Sargassum whitey in the synthesis of nanoparticles. While using, first, this work was carried out in the year 2007. I was part of this research. While just synthesizing, taking a bulk mass of Sargassum whitey, the extract and just used with chlororic acid. we could find that the reaction took place after 1 month. But today, in the last few years of various other researches, you would have just seen where we can just synthesize an nanomaterial within 10 minutes, 30 minutes. This is the first report which we carried out using marine algae in way back in 2006, where it took nearly 120 hours. That is the stability. So even similarly, the stability of the nanomaterials was also the same when recording the even after 2 months and 6 months also when we just checked them. These are the same images which we recorded for the Sargassum nanomaterials. It was just a precursor reduction. There was no heating, no refluxing. So the experiment was carried out at the room temperature itself. This was the article in which we published that paper, where it was published in Collections Surface B. Here, this is the first report, as I said. Marine algae can be used for synthesis of nanoparticles and gold nanoparticles. And after synthesizing gold, we move to silver also. In silver nanoparticles, as I said earlier, if it is going to be a gold nanomaterial, it will be having a wavelength at 520 nanometers. If it is going to be silver, it will have its wavelength at 420. Again, you can just vary the plus or minus 20 nanometers of change in the wavelength for the materials. Okay, so using this silver nanomaterial, we synthesize the silver nanoparticles using Sargassum YT. It was published in Journal of Nanoscience and Nanotechnology. In this, we just initiated the antibacterial studies also, where you can find out that the reductant, if for example, using FAR, a simple thing which it can help us is we can identify the functional groups which has been involved. If it is going to be a thiol, you will be getting a peak around 2005 or something. If it is going to be a carbonyl, you will be just trying to have a peak around 1700. So if there is a change, if there is a shift in the peak before the extraction, after the synthesis, we can just try to narrow down that the carboxyl group has involved or a carbon group has involved in the stabilizing or in synthesis of this nanomaterial. We just also carried out a small antibacterial activity studies in the way back 2008. And it was published in Journal of Nanoscience and Nanotechnology. And we also Sargassum variety, we synthesized zirconia nanoparticles, and we just studied for the antibacterial activity. We can just see if it is going to be a gold study, it will be having its particular peak at 38, 44. These are the we having. And as I said, for gold, it is going to be 520, or for silver, it's going to be 420. For zirconia, it will be around 290. Okay, so using a UV spectroscopy itself, you can just come to a conclusion that yes, we have just arrived at the result where we could just find a shift in the UV spectroscopy. Okay. Ideas of the research was we just this is a bacterial part of work where the synthesis of the nanomaterials was just carried out, and we just studied the zone of inhibition also. So using Sargassum YT, we have just carried out this gold, silver, and this zirconia synthesis also. And this was published in the journal Microbial Pathogenesis. And yet another work, this work was carried out in association with the CAS Marine Biology. Professor Sir was part of this work, and it is his work. And I was just supporting him in synthesis part. And this particular synthesis, they just studied the efficiency of the gold nanoparticle and its antibacterial activity, where we can just find out that nano triangles were formed. Generally, we can just only polydispersed nanomaterials. We could, in biosynthesis, we couldn't come across easily of this nano triangles and all. And in this particular work, we just got the nano triangles, and it was published in association with CAS Marine Biology. This is the work. It was published in Spectroscopy Acta. And one more paper, this is our work. Sargassum myriocystum we collected in Mandagum. We just synthesized this nanomaterial. We could find that the stability of the Sargassum myriocystum synthesized nanoparticles was same when we even after the 3 months or even 6 till more than 6 months, we could find that these nanomaterials were stable. And we're just working on other catalytic application also for this work. And here, I would just like to make one more point very clear. We would have just read about the Tyndall effect. to just make it more clear, you just imagine if a colored solution can't be called as a nano solution. Okay, we just know that a laser light can pass through, or a light can pass through a particle. Okay, so here I would just want to give that information. You just take a small vial, you just have a 100 ml of chloride acid. You just use this laser light. You can see that the laser light will not pass on the other side. Okay, since it is not in the nano level. Okay, but if you just take, add your reductant, and you ensure that you are getting a rubin with the wavelength from 500 to 540 nanometer. You can just see that. You can see that when you just pass this laser light, the laser light will be passing on the other side. With this, you can infer that the particles are individual, and it is permitting the entry of light to the other side. This is the difference between oxidation state, where we were talking about chloroauric acid, and the reductant nanoparticle. Okay, this is the image of the Sargassum myriocystum. This is also one of the mode to just identify. For example, if you're just trying to synthesize, and if you just take a color solution and tell, sir, I just have this nanomaterial, it can't be accepted. So Sargassum myriocystum, and in this study, we just identified that a cyclopentyl derivative compound was there. After, as I said, it was just a step-by-step carry forward only. In 2008 and 9, when we just synthesizing, it was just to identify whether we have a nanomaterial. Yes, we have a nanomaterial identified. Next, we're just trying to apply it for a catalytic study, or we're just trying to work on various other biomedical applications. Now, the step comes forward where we are just what has reduced, what has exactly involved. So, in that search, we could just identify that Sargassum in myosystems, a pentyl derivative compound was there in the synthesis of this nanomaterial, and this we identified using GCMS. And this is the paper which we published. And apart from that, we also synthesized silver chloride nanoparticles used in marine algae, and we studied its antibacterial efficiency. Here we just used the Sargassum plagiophyllum. We were just using various Sargassum species, Sargassum whitey. We were just using Sargassum myriocystum, plagiophyllum. So we were just using different. In this case, it is not that all the algae are reducing all, or all the fungus are reducing all. In case of biosynthesis, if you want any back reference, I would just suggest you can just check Dr. Murali Shastri's web page or his research, where the biosynthesis, how it occurs. He is the pioneer in this, since he started this research in biosynthesis in 2003. So based on that, we were just trying to identify how the biosynthesis. So it is not that you take the auric chloride, if all the algae will be reducing all the cells. No, there are different in the collection spots also. When you just collect the same Sargassum whitey in the coastal part of the Andhra, and you just take, there also we could find some reduction. So the next step is, excuse me. So the next step is to identify the capping molecule, or to identify the molecule which can stabilize the synthesis. So we just synthesized the silver chloride nanoparticles. It was having a wavelength of 410 nanometers. And this is the XRD. Here in XRD, you can see that earlier we used to call it as a JCPDSS file. Now it's called ICMM. It has just changed. So while recording XRD, it was identified that, and in this particular work, I would like to thank Dr. Ramachandran. He was the one who just identified, and repeated studies also we could identify that the reductant formed, the nanomaterial form was silver chloride, and we published this article in Spectroscopy character. And now from algae, since our study, since we're talking about the synthesis of from marine sources, now we're just moving to algae to sponge. This particular was carried out in our center by Dr. Indranath sir, my HOD. He was just using Actinella elongata. Here we can just see marine sponges are considered source of all chemical molecules, since they are withstanding various chemical environments. Now we couldn't work on sponge that much easily. We need special permissions. But marine sponges are having huge chemical potential. And in that case, when we were just trying to work on this, we just reduced chlororic acid precursor using the sponge extract. You can see the XRD. You can see that the difference between the particular Sargassum and Facile and gold and this particular work, the particles which are synthesized are monodispersed in the entire thing. That is the very important thing for this. That is what the search is all about. We're just trying to find out which has potential. No, if I could save this extract, can I get the more study? Yes. Then alone, on the trial and error basis alone, we can come to a conclusion. Yes, we are just getting a more synthesis. But in case of Sargassum waiting, we got polydispersed only. Monodispersed was less. And he published this work in Colloidal Surface B using marine sponge synthesis. Beneficial bioapplications of cellular nanoparticles synthesized by marine crustacean. For earlier, I just said an example of marine sponge. Now I'm just taking an example of marine crustacean. This particular work is not carried out by our institution. It was carried out in Egypt. I just want to show you that the resource or the potential which are available from marine environment are being utilized in various parts. So the search can be extended further also. So one of the researchers in Egypt has just carried out the synthesis of cellular nanoparticle using marine crustacean. What is of this thing is they are just carried out to the synthesis and they are just compared with the control other thing. This particular cellular nanoparticles was just answering the antiviral, anti-diabetic, anti-arthritic, anti-aging, and anti-inflammatory related studies. And that is why this particular study I just shown, they have just published this journal. So even marine crustacean and marine sponge, marine algae, all these things are just trying to say to that we have to just identify the novel chemical molecule in them. Okay, this is the paper which they have published. And this is some other work which is not related to our research, but I want to show you that the extracellular synthesis of zinc oxide nanoparticle has been carried out, where I am just trying to move from gold, silver, noble metals to metal oxides. So metal oxides are more stable in all the environments. So there is a possibility to synthesize the nanomaterials using seaweeds. One more work is iron oxide. So the earlier you saw is zinc oxide was one metal oxide. The other one is brown sleeve, Sargassum actinum. This particular study was also not from India, but I am just trying to bring to attention that the synthesis of iron oxide nanoparticles, zinc oxide nanoparticles are also being carried out using Sargassum species. And the intracellular and extracellular synthesis of nanoparticles. Here, it's just information to understand how this happens within the cell and outside cell. So most of the synthesis we just carried out, which we just take forward are extracellular only. We just take the extract, we just crush and just trying to identify. And same applications and the stability is high when some studies are being carried out in intracellular also. So this particular image has been taken from biomolecules, and this is how the reduction when your precursor if you just take it for synthesis of gold nanoparticles, the precursor just goes forward. You can see to that the growth phase, the proteins, polysaccharides, all which are involved within the cell, they just reduce, and they just give additional property, which I just explained in the last catalytic example. Which I want to say is all are silver, all are gold, all are palladium. Then what is the necessity arising to vary the nanomaterial? That is where the intracellular synthesis gains. The metabolites secreted or the presence of the polysaccharides are varying the property in application studies. So that is why the intracellular and extracellular synthesis is gaining its importance. Showing the understanding how these studies are being carried out. And these are the various studies which are reported from, all the studies are reported from India itself. And you can see these are the various marine bacteria which are just used to synthesize the semiconducting nanomaterials, gold, silver, manganese oxide. All these are used in this particular studies. And in case of the synthesis of marine bacteria, and again you can see aspergillus, penicillium femtinum, they are used to synthesize this gold and silver in this marine fungi case. In application of marine source derived nanomaterial, here we will just discuss 2-3 examples. I'll just try to explain how the capping molecule or how the chemical molecule of the particular resource is just enhancing the property. This is what I will just try to explain here. Marine fungus mediated cellular nanoparticles. This particular work was carried out by Mr. Fayaz, and one of the author was from Satyabama Institute of Science and Technology. And this particular paper is the topmost cited paper of Satyabama Institute of Science and Technology. This has nearly 1,000 citations. This is the highest cited article, and it is from School of Bioengineering. Okay, here the research activity is he has just taken a marine fungus. He has just used to synthesize the cellular nanoparticle. What he has done is he has just studied the synergistic effect. Okay, when you just synthesize the nanoparticle, cellular nanoparticles in a conventional method, and you just try, we could see to that the effect of the antibacterial activities along with the antibiotics of kanamycin, erythromycin, and chloramphenicol and ampicillin was increasing. You can see the graph which I have given there. This is the difference, or this is the potential of that marine fungus. So that marine fungus has the capability to improve the activity of the antibiotic. That's what it has mentioned as synergistic effect. So the search ends here, where we can just see to that, visualize that in extracellular synthesis, when you just synthesize the properties in some cases where the marine fungus has the potential, you can see that there is an enhanced activity. And in case of the second study, this is about a nano bioremediation. Here, what they just used is the alga lacta. They are just used to synthesize the cellular nanoparticles, and here methyl orange degradation. This is what the study is all about. So when you just carry out this alga lacta in the photocatalyst condition, in the exposure of light, you can see to that there is a degradation in the studies. There is a degradation of the methyl orange color. So the dye degradation is occurring when this particular report was given. This alga lacta has the capability, the chemicals present in this has the capability to stabilize the nanoparticles, and it was involved in the application. Third, a catalytic application I have just given here, apart from the dye degradation. Here, in case of this biosynthesis of palladium and gold nanoparticles, they are synthesized using Facile species. And this 4-nitrophenol is one among the important chemical to be noted, as it is used in the manufacture of drugs, fungicides, insecticides, dyes, and to darken the leather. So the reduction of this 4-nitrophenol is very important. So when you just synthesize this particular nanomaterial, they are just to see that it can, the catalytic property of this Bacillus species synthesized nanoparticles, we're just trying to convert into aminophenol. So the reduction property, so where you are just trying to divert a toxic chemical in the environment to a less toxic chemical, is the first initiative to stop the toxic effect. So this is what they are just studying, a catalytic application. In case of antifouling agent, it was just again one among the study where I just come across this paper. The Turbinaria ornata was just used to synthesize the cellular nanoparticles, and they are just tried to isolate 15 different biofilms, and they could see that the efficiency was nearly 71%. So the antifouling effect or the catalytic effect or the dye degradation studies, some selective molecules are showing the activity. Though the research is in progress, we know this antibiotic, and in case of antifouling, we just those IMO, the Civil Maritime Organization, has banned this activity. So there is a long, very big search for this antifouling activity, and this is in progress. And other applications are anti-cancer studies, anti-diabetic studies, where they're just trying to reduce that. These works are in progress. In case of agricultural-related disease management, we can just say that the iron deficiency, iron deficient crops, they're just trying to use the nanomaterials which are synthesized from the seed species, and to increase the yield and disease management also they're just trying to use. In aquaculture disease management, gold nanoparticles are just being used to control. These sort of studies are in progress. And with this, I just end my lecture, and I wish to express my sincere thanks to the Managing Department of Technology, Direct Research Madam, Head Center for Ocean Research, Head Department of Biotechnology, my research scholars, and the entire team Ocean for their support. This is my mail ID. If you have any queries, if I could answer, you can just write to me. Thank you all.
Thank you, sir. Thank you for the wonderful presentation that you have given. Participants, if you have any queries related to the topic, you can ask in the chat box, or you can raise your hand so that I can unmute you. You can communicate with the speaker right away. So this is this 5 minutes is for the interaction session.
Actually, Vinith, I just want to give one announcement here.
Yes.
Actually, I saw some of the messages in chat box. They want the soft copy of the PPT. Actually, this recording will be in the YouTube soon, and the link will be shared to all the participants. They can view the YouTube link soon. That is the information to all the participants here.
Okay, so thank you, sir. Participants, if you have any queries related to the topic, you can directly ask in the 5 minutes which we have left now. Or you can chat, you can give a question in the chat box also. Yes, sir, we have a raised hand. So,
yes, sir. Yes, please.
Yes, ma'am, please go ahead.
Yes, sir. Regarding the synthesis of nanoparticles, sir.
Yes,
You have mentioned about the marine crustaceans. Only
yes,
that species are involved in a synthesis, or any other species also involved in synthesis of nanoparticles, sir? Why specifically the marine crustaceans and sponges?
No, ma'am. Actually, marine sponge was a part of the research work in our research center, ma'am. Next, Dr. Inbakandan sir is the corresponding author, and it is his work. We were just trying to bring it to the attention of all the speakers that marine sponges, marine crustaceans, marine algae, marine mangroves, all are involved in the synthesis. That the search is to identify the chemical molecules which can enhance the catalytic or the biomedical application. Marine crustacean, it was just a study which was published in 2021 recently, and it is just a research work which is being carried out in Egypt, and they were just trying to bring it to the attention to the audience that marine crustacean extracts are being used in the synthesis, and it is having the anti-diabetic and antiviral properties. So it is just the potential of the chemical.
So then the lower invertebrates may also be involved, no, sir?
Yes, ma'am. It will be involved.
Like,
it will be. Yes, ma'am. It will be involved.
If we are interested, we can also take.
You can work on it.
Yes.
It is wide open. That is what that is what the main focus of this talk, ma'am. So it's wide open. So you can just try.
Because there was a. Because there was a saying that only in plant extract we can synthesis this nanoparticles. That's what I am asking this question.
I am not talking as a ocean research scientist, but I am just giving a general comment, ma'am. The one of the PPT I just shown, the stability of the seaweed molecules or the marine sponges is high because it's withstanding the varying pH or varying temperature or varying environment, ma'am. So the stability of the synthesis from the marine source is much better than the plant synthesis, ma'am. That is what. So you can try, ma'am. Thank you.
Okay, sir. Thank you, sir. Thank you.
Thanks, ma'am.
Thank you, ma'am. Participants, if you have any queries, please kindly raise your hand and communicate with the speaker. Yes, sir. Do you have any questions, sir? Chris Roy, sir?
Yeah.
I'd
like to thank the speaker first of all for the very simple, but he knows what he's doing. Really like to thank him for that, and it's really nice to see the facilities that you have built up over the years. Looks like you started everything from scratch.
Yes.
So at Emmet, we are actually looking forward to learning from you, if I could put it that way. Yeah, so I'm going to use you as a template, if you don't mind, Inba. I'm going to use you as a template. Okay, but we'll try to build something unique, probably together. Okay, so I'm looking forward to that, and I have lots of clarifications which I think I'd rather come in person and do it. So we have a nice team here, and we'll come in person to Satyabama for sure. And I really like to thank you for making it so lucid. Thank you so much, Dr. Ganesh.
Thank you, sir.
Thank you, sir. Thank you for the kind words, sir. And sir, the Milton Prabhu, sir, you can unmute yourself and you can clarify your doubts, sir.
Yes, good evening, sir.
Good evening, sir.
Sir, the presentation was very nice. The Ganesh Kumar sir showed the presentation regarding that synthesis as well as the techniques that we can use to characterize the molecules and all. Everything is very nice. And I really congratulate Dr. Impakandan as well as the team in your they are doing very great expertise in this particular area, and the facilities you have built up, that was very nice. And my clarifications regarding your presentation, I am having one simple doubt regarding the application of these particular nano materials. So far we have used a large group of marine sources for the synthesis of these particular nanomaterials, and most probably that metal particles. So what should be these particles can produce as the impact on the organisms in bio? Is there any toxic effect has been reported? Because there are a large number of toxic impact has to be produced nowadays regarding the impact of these nanomaterials when we are using that in vivo system. So what is your opinion regarding in these applications of these nanoparticles?
So in case of in vivo or the metal sector, the importance I was just trying to emphasize is basically in biosynthesis, since the compounds which are going to be there in the seaweeds or in case of the marine fungi or the secondary metabolites, they will be having less impact than those of the chemical molecules. For example, if you're just trying to take a citrate, or if you're just trying to take any chemical molecule, the degradability will be high if it is going to be a chemical molecule. But in case of the biosynthesis source, the impact will be lesser. The biodegradability possibility will be high. That is what the importance is.
Sure. Thank you very much.
Thank you, sir.
Thank you, sir. With this question, one more question is there in the chat box, sir. With the question, we can end the session, sir. And Ramya ma'am is asking that whether only the extract is involved in biosynthesis of nanoparticle, or the living organism itself can be used.
Yes, ma'am, that's what the difference of intracellular and extracellular is trying to explain. The extract, if you just take, if it is going to be seaweed extract, it's going to be extracellular synthesis, where the surface molecule and the attachment. If it is going to be a living cell, that's where the intracellular concept arises. So we can optimize where when we're just trying to explain about the application. In case of intracellular nanomaterials, we are just trying to show that the reduction will be having the impact of polysaccharides and other chemical molecules which are present in the cell. So both will have its impact, ma'am. That will be deciding the nucleation also, and that will be having the impact on the size and its shape also. So both are entirely different. Both will have its impact.
Okay, sir. Thank you, sir. Thank you so much. That's all from the participants.
Thank you,
sir.
Thank you.
Thank you, sir.
Actually, thank you, Ganesh sir. Actually, to be frank, to the all audience, Ganesh sir is not feeling well for the past days. Because for my request, he is accepted and he is ready to give this first talk. Because we all learned the nanotechnology from Dr. Ganesh only, and that's what I thought. If he is giving a talk on the initial stage, it will be very useful for everyone to know what is the basic of nanotechnology. And thank you, sir. As I already used to say that it is very crisp and clear. Thank you. Thanks a lot, sir.
Thank you. Thank you, sir. Thank you all. Thank you.